So previously we have looked at the innate immunity, which remember is our non-specific immunity that we're all born with. It is not unique to any person necessarily. But our adaptive immunity is a specific immune system that is trained. And so when we are born, we do not have much of an adaptive immune system, but we do have all of our innate immunity. But we are going to train our adaptive immune system to recognize pathogens. Do this with vaccines and natural exposures. So with our adaptive immune system, we have three main cells. We have our CD4 and our CD8 cells, which are lymphocytes. And then we have our B cells, which are also lymphocytes. So the T cells are going to be our cell-mediated immunity. The T helper cells will express CD4, so a cluster of differentiation 4. And cytotoxic T cells express a CD8. They will respond primarily to cells infected with intracellular pathogens, so things that are within the cell, such as viruses and intracellular bacteria. Cancer cells, because cancer cells have mutated, are no longer self. And foreign cells, such as those that are transplanted. So when you have a transplant rejection, it is usually due to this cell-mediated response by the T cells. T cells are made in the bone marrow. They leave the bone marrow, go to the thymus, where they are stimulated to mature, and if it is self-reactive, it will be destroyed. Once are mature, naive, so they're mature, but they have not been exposed to antigen yet, so they are still considered naive, will migrate to the lymph nodes until they encounter an antigen-presenting cell, which will activate them from being naive to a mature T cell that can combat whatever pathogen was presented by that particular antigen-presenting cell. And so only certain antigens are called immunogens, but they're capable of eliciting a response from our immune system. Antigens that are not able to do that are called haptens. So these are very small, and they have to be attached to a carrier protein to elicit an immune response. The antigens that are present on your own cells are called self-antigens, and you do not want your immune system to react to those. If it does, then you have developed an autoimmune disease. And so the antigen will bind to an antigen binding site. It is very specific, lock and key, but you have to have a cell to become activated to the antigen before this will happen. And so that particular antigen that will do that particular portion is known as our antigenic determinant. And so these antigens that we have been talking about are presented via our major histocompatibility complexes. So antigens bound to glycoproteins, called the major histocompatibility complexes, are presented on cell surfaces to our immune system, and that will enable our immune system to become active. And the MHC came from the fact that it is a major determinant of the compatibility among tissues and organ donors for recipients onto whether things can be transferred between individuals. You find MHC molecules on nearly all nucleated cells except your erythrocytes. So you're going to find them on all nucleated cells except erythrocytes. And so to make blood products considerably easier to donate than other organs and tissues. And so we don't have to do as much screening for blood other than for our blood types, which we talked about back when we talked about blood in chapter 19. And so those MHC are basically docking sites for the antigens to be displayed. So here you can see with MHC1, it is going to present endogenous antigen. So the antigen is within the cell, made within the cell. It is degraded, put through the rough endoplasmic reticulum where it is docked with an MHC class 1. Then once it is docked with an MHC class 1, goes through the Golgi and then presented on the cell surface to help activate the immune system. Whereas our MHC class 2 presents exogenous, so things that are taken in, so something like a bacteria that is engulfed is then broken down. The antigen is put into an MHC class 1 in our rough endoplasmic reticulum, processed through the Golgi, and then is displayed on the outside of the cell. And so here is a summary of what we just talked about. Class 1, found on the surface of plasma membrane of nearly all nucleated cells, presents endogenous antigens, or those that are synthesized inside the cell. Cytotoxic T cells are the ones that interact with MHC1, and it will only interact with MHC1. Whereas our class 2 are found only on the surface of antigen-presenting cells. And so the class 2 antigen-presenting cells present to the T helper cell and then activate our T helper cell. Class 2 present exogenous antigens, or those cells, those the cell takes in via phagocytosis. So the exogenous originates outside the cell, must be taken into the cell by phagocytosis, and then added to the MHC complex. Whereas endogenous is either a foreign antigen present on a pathogen that lives inside your cell, such as those intracellular bacterium, a virus. If your cancer cell, because your DNA is mutated, so it is no longer a self-antigen, these would all be considered endogenous antigens. Endogenous can be foreign or self, because if it's an intercellular bacterium or viral, then it's foreign, but if it's a cancer cell, it is self, whereas our exogenous are almost always foreign. So why do we need both? Well, we need both because when the cytotoxic T cells are going to recognize an endogenous foreign molecule, that means that that cell itself needs to be destroyed. And so the cytotoxic T cell will destroy any non-self antigen presented on the MHC1 and destroy the cell itself. But with MHC2, it is presenting exogenous, and the antigen-presenting cells themselves are not diseased. They have taken in a foreign particle or are destroying it and then presenting it to the immune system. So you don't want to kill your antigen-presenting cells. So those things that are exogenous, presented on an MHC2, are a warning system. They're saying, hey, this is in here, mount a response, activate our B cells, activate our cytotoxic T cells, but mainly our B cells. So you don't want to kill the messenger here, because all it did was clean up the garbage or phagocytize whatever the foreign invader was and then present it to the immune system. And so here you see the cytotoxic T cell. Like I said, it is presenting endogenous antigen, means that the cell itself is infected or diseased or mutated. And so you want to kill that cell because whatever is inside of it is causing issues and is foreign and is going to possibly kill the whole organism, us. And so that's why MHC1, endogenous, stimulates the cytotoxic T cell to kill the cell that is presenting it. All right, so here is the figure in your book depicting the class 1. And so the cell synthesizes self-antigens, the antigen broken down. The fragments are paired with a MHC1, that antigen fragment transported to the rough endoplasmic reticulum where that coupling takes place. And then the vesicle carrying the antigen and the MHC1 displayed on the cell surface. And the cytotoxic T cells can come around and say, oh, is it self or is it foreign? If it's self, we ignore it. If it's foreign, then we kill the cell. Whereas MHC2 presents exogenous antigen. So that is something the phagocytic cell takes in an antigen, joins with the lysosome, breaks it down. Then those fragments are going to be joined with the MHC2, which is coming out of our rough endoplasmic reticulum. Those two vesicles fuse together. When the vesicles fuse together, then the antigen is put into the MHC2, presented on the cell surface, and then that is going to activate the T helper cells, and the T helper cells are the linchpin to the immune system. So they are going to further amplify our B cells and our cytotoxic T cells by releasing cytokines that we talked about in the previous lecture on innate immunity. And so how are the antigen presenting cells going to do this? So one of our big antigen presenting cells are our dendritic cells. Dendritic cells display the antigen fragments on their MHC. They present both cytotoxic MHC1 and the T helper class 2. And when they are binding to the dendritic cell, they are naive. And then once they encounter the antigen, then they are stimulated to differentiate and become mature, and they create clones and memory cells. Once they have been activated, and a way that the body prevents accidental activation is that the dendritic cells have a co-stimulator, and the other cells in the body don't have this co-stimulator, so the cytotoxic T cell has to see the MHC class 1 and the co-stimulator. Same with the T helper cells, has to see the MHC2 and the co-stimulator to be activated and pushed into creating clones of themselves, actively dividing. Those actively dividing cells, the majority will become what we call effector cells, but a few will be pulled and put into memory. So that's when we get a stronger response the next time we see that antigen, is because we have memory cells that are already primed to recognize it. And so here we can see in the lymph node that another type of antigen presenting cell presents it on an MHC, the T cell binds, co-stimulator. So this is just a generic version of what we saw with the dendritic cell. Stimulates them to bind, effector cells, and memory cells generated from that activation. And so when we say the T helper cell is activated, it releases cytokines. Some of the main cytokines are interleukin 3, interleukin 2, other interleukins. And remember, interleukins are 29 of them, I think. And they just keep numbering them. And then the T helper cell directly stimulates the B cells to proliferate. So interleukin 3 is going to enhance the innate immunity. by enhancing phagocytosis and by triggering the macrophages to make interleukin 12, which then stimulates T helper cells and creates kind of like a positive feedback loop. Interleukin 2, activation and proliferation of cytotoxic T cells, and all the other various interleukins are going to stimulate B cells to proliferate and make antibodies. So these are all ways in which the T helper cell, or linchpin of the immunity system, is able to enhance and activate the other two arms of the immune system, our cytotoxic T cells and our B cells. All right, so let's watch this big picture animation on activation of helper T cells.
Infection with the virus that causes the common cold activates multiple innate immunity responses, including inflammatory mediators, interferons, and natural killer cells. Another component of innate immunity that gets activated is the phagocytic cells, such as dendritic cells and macrophages. Local or resident dendritic cells ingest pathogens, cellular debris, and even intact infected cells by phagocytosis. These dendritic cells, along with viral particles, then travel through the lymph to lymphatic organs and tissues, such as the lymph nodes. There they present the antigens they have previously ingested and processed into fragments to lymphocytes, such as helper T cells. A naive helper T cell becomes activated when its T cell receptor binds to a dendritic cell's class II MHC molecule presenting viral fragments. To become fully active, the helper T cell must also bind a co-stimulator surface molecule, which prevents naive helper T cells from becoming inappropriately activated. The activation of the particular naive helper T cell that can recognize the class II MHC antigen complex is called clonal selection. Clonal selection is followed by helper T cell proliferation. Multiple copies of the activated cell, which are all capable of recognizing the same virus, are produced by cell division. The army of activated helper T cells then become effector cells. They secrete cytokines, which are essential for activating naive B cells and naive cytotoxic T cells, crucial components of the adaptive immune response. Finally, some of the activated helper T cells differentiate into memory T cells to become part of our immunological memory. If we encounter the same antigen in the future, these memory helper T cells mount a quick and efficient response, so we rarely experience any symptoms. However, if the antigenic fragment of the virus mutates, the memory helper T cells will not be able to recognize it, and the adaptive immune response will have to develop from the beginning.
So the effects of our cytotoxic T cells, activated in the same way, they are going to bind to the MHC class I instead of the MHC class II. Once they bind to that MHC class I presenting the foreign antigen, remember it's going to be endogenous, made within the cell, meaning I need to kill this cell because it is infected with whatever I don't like. It will do the same thing as our natural killer cells. It will put in perforin proteins, secrete enzymes into the cell that will destroy the cell's DNA, causing the cell to degrade. But an added layer of security with our cytotoxic T cells, since they do kill the cell, is that they also require the interleukin 2 from our T helper cells. Remember, we saw that a few slides back. And so the interleukin 2 is just another layer of protective mechanism to prevent the C cells from killing good cells for them to be activated fully.
The primary function of cytotoxic T cells is to kill abnormal cells, such as those infected by viruses. Cytotoxic T cells become activated in lymphatic tissue by recognizing antigens presented by dendritic cells, a process called clonal selection. Dendritic cells display class I MHC molecules in addition to their class II MHC molecules we discussed in one of the previous animations. A naive cytotoxic T cell becomes activated when its T cell receptor binds to the dendritic cell's class I MHC antigen complex. This process requires a co-stimulator and interleukin-2 from helper T cells to prevent unnecessary naive cytotoxic T cell activation. As with other lymphocytes, the step that follows clonal selection and activation is proliferation. The activated T cell clone divides and gives rise to many identical effector cytotoxic T cells. Next, the effector cytotoxic T cells do their job of recognizing and destroying the infected host cells. Infected host cells display viral antigens on their class I MHC surface molecules. Cytotoxic T cells recognize these antigens using their T cell receptors and bind to the infected host cells. Like natural killer cells, effector cytotoxic T cells release perforin and then release enzymes into the cell. Here, the enzymes catalyze reactions that fragment the cell's DNA and lead to its death. Finally, some of the cytotoxic T cell clones will differentiate into memory T cells to ensure immunological memory. Some memory cells tend to live longer than others. Memory cells for the common cold virus stay in the circulation for a few weeks or months after the infection, but eventually die off. This makes us more susceptible to infection again.